Over winter break, one student from MSJ traveled to Africa and unfortunately came down with Lassa virus while coming into contact with the vector multi-mammoth rat. Lassa virus is a single-stranded RNA virus that was discovered in 1969 when a Nigerian nurse became infected. The virus itself, however, is a zoonotic virus which originates in the multi-mammoth rat, targeting other animals, and humans are accidental hosts. Fortunately, Lassa fever is generally mild or has no observable symptoms in about 80% of the people it infects. The virus is widespread in West Africa. Nearly 5,000 people die from this virus each year. Specifically, Lassa virus is found in the Mano River region, including countries such as Nigeria, Liberia, Guinea, and Sierra Leone. Lassa virus predominantly occurs during dry seasons. Since the African climate is normally dry, the disease is relatively constant throughout the year. To date, in the United States, there have only been 2 cases of Lassa virus, both of which occurred due to American travelers in the infected area of Africa. As experts of the Lassa virus, we are concerned about the health of students at MSJ and are afraid it is going to spread.
Once a person has come down with Lassa fever, the disease can be spread through direct contact with infected body fluids, such as the blood, urine, and vomit. The disease enters students through the bloodstream, lymph vessels, respiratory tract, or digestive tract. Once inside the body, the virus multiplies inside cells. The replication causes capillary lesions, which in turn causes platelet loss and tendency towards bleeding. Capillary lesions also cause increased vascular permeability and hemorrhage in many organs like the stomach, kidney, lungs, brain, and small intestine. Common symptoms include fever, myalgia or muscle pain, decreased energy, sore throat, edema, rash, and gastrointestinal complications. If one is lucky enough to survive this fever, the symptoms persist for 2 or 3 weeks, usually subsiding with the onset of deafness.
If you experience any of these symptoms, go to the office and contact your doctor immediately. In order to determine if you have Lassa fever, you will undergo a variety of tests because the symptoms of Lassa virus are often confused with those of malaria, as well as other viruses in the same arena virus infectious disease group. Lassa virus is similar to other arena viruses since the whole group causes viral hemorrhagic fevers, also known as VHFs. All VHFs affect the vascular system and negatively impacts many of the body systems. Some tests you will undergo include albuminuria, which is testing for the presence of albumin in urine, reverse transcription polymerase chain reaction, aspartate aminotransferase test, also known as AST, and alanine aminotransferase, which is also called ALT. AST and ALT can help determine the extent of liver damage by measuring the amount of either aspartate aminotransferase or alanine aminotransferase in the blood. A high presence of either of these enzymes is a strong sign that you have Lassa fever. Also, enzyme-linked immunosorbent assays, also known as ELISAS, can help detect viral antibodies. These enzymes can detect immunoglobulins, such as IgM and IgG.
If you're diagnosed with Lassa fever, you may be given injection of antiviral drugs, ribavirin. Ribavirin inhibits RNA-dependent nucleic acid synthesis, which greatly alleviates many of the symptoms caused by RNA-stranded Lassa virus. Of course, you will still need to receive supportive care from maintaining fluids, oxygenation, and blood pressure. Unfortunately, as of now, there's no vaccine for humans. Scientists, however, are coming close to eliminating Lassa virus from human race with their successful vaccination of guinea pigs and non-human primates. To prevent the spread of this illness, it is crucial that basic precautionary methods are used as abstaining, contacting body fluids, and frequent hand washing. Mousetraps should be also placed at homes. The public can also fight this disease by educating those in high-risk area about ways to decrease rodent population in their homes. This will greatly help control and prevent further cases of Lassa virus.